Before we get started on the day-to-day? It's early, you guys are quiet. Okay, so there's no questions. Let's go ahead and kind of jump into things. So on Friday, we had gone over or introduced the autonomic nervous system. And in the lab, we started to talk a little bit about the different sympathetic pathways, right? But we had a couple of different scenarios here. We had a somatic motor pathway, somatic motor, right? Where we had a neuron in the central nervous system. This is our CNS right here. We had a neuron right here. I think this is in your notes. And we had a neuron going to a target, which is a skeletal muscle. We had a situation that kind of looked like that. And then we had parasympathetic motor. Again, arising in the central nervous system. And we had a preganglionic neuron, a little bit bigger, preganglionic neuron synapsing with a postganglionic neuron going to some sort of a distant target, right? And then we had a sympathetic motor. We had a sympathetic motor, again, CNS origins, and again, preganglionic neuron synapsing with a postganglionic neuron. Again, going to our distant target. Right, so we're going to our distant target over here. We kind of left things here on Friday. Obviously, there was a little bit more to it. One of the things that we said is that the sympathetic and the parasympathetic motors differ in a couple of different ways. One of them is their CNS origin. So what's the CNS origin of our parasympathetic motor pathways? Where do they arise in the central nervous system or start? Good thing we're not having a quiz this time. Where does parasympathetic neurons, where do they start in the central nervous system?
Do you mean like what part of the spinal cord?
Yeah.
The ventral?
Nope.
Okay.
Where do they start in the spinal cord, or where do they start in the central nervous system? Some of them start in the spinal cord.
The nerves, the finger nerves?
Which ones?
3, 7, 9, 10.
3, 7, 9, and 10, and where else?
S2.
S2 to S4, right? So the sacral region, right? Remember, our parasympathetic motor pathways have a craniosacral origin, right? And then sympathetic motor pathways, where do they start? Thoracic, right? T1 actually to L2. We'll generalize it though, and we'll say the thoracic region, okay? So craniosacral origins, thoracic origins, right? The other way in which they sort of differ anatomically anyway is the location of the ganglia. I kind of portrayed that here. Remember that with the sympathetic motor pathways, the ganglion is really quite close to the CNS usually. So we have a fairly short preganglionic, fairly long postganglionic neuron, whereas it's kind of the opposite in parasympathetic. That ganglion tends to be a little bit closer to the target tissue. Now, obviously, the idea goes a little bit farther here. We had talked in lab on Friday about the sympathetic motor pathways, and we talked about how those ganglia primarily exist is that chain of ganglia that exists right next to the spinal cord. In fact, your guys' homework over the weekend was to practice those pathways through the sympathetic chain using the white and the gray ramus communicans. And we'll talk a little bit more about that this morning in lab, and I'll give you guys an opportunity to ask questions and things like that. But for today, this works. There's one final autonomic motor pathway that we haven't mentioned yet, but we need to sort of bring it up right now. And that one is what we call the adrenal sympathetic pathway. And it involves, as the name implies, our adrenal glands. So let's go ahead and bring up our one final here. This is the adrenal sympathetic. The adrenal sympathetic pathway. Again, we're looking at CNS origination, and just like with our other sympathetic pathways, we're looking at T1 to L2, so that doesn't differ a whole lot. Okay. But we have to consider the anatomy of the adrenal gland here in order to really understand this. So, in your abdomen, your kidneys sit in the very back of your abdominal cavity. Have two of them, probably, and they sit in the back of your abdominal cavity. And on top of your kidneys, you have these two small glands called your adrenal glands. So they kind of sit, they're kind of cute actually, but they sit on top of your kidneys like little stocking caps or tubes. They sit up there like this. So this is an adrenal gland right there. Probably today, and if not today, on Friday, we're going to transition to start talking about the endocrine system a little bit. This is actually going to be our segue into that. And when we do that, we'll look at the anatomy of the adrenal gland in a little bit more detail. But suffice it to say for right now, the adrenal gland does sit on top of the kidney, and there's actually two layers to it. So there's an outer layer, and then there's an inner layer. So it looks kind of like this. If we were to kind of slice it in half, we'd see that there are two layers. The outer layer is what we call the adrenal cortex. And the inner layer is what we call the adrenal medulla. So we have these two primary layers to the adrenal gland. They do different things. Again, and if not today, by Friday, we'll definitely kind of circle back to the adrenal gland a little bit more properly. But the adrenal sympathetic pathway involves that inner part of the medulla, or that inner part of the adrenal gland, the medulla. Okay, so let's take a really quick look at how this guy sort of looks, and then we'll kind of start to talk about how these pathways actually work a little bit more. So again, we're looking with the adrenal sympathetic pathway, we're still looking at a T1 to L2 origin. So it's no different than this one in terms of where it originates. And the neurons are going to use the sympathetic chain in the same way. They're going to enter via the white ramus communicans, and then they're going to go like down one of the splanchnic nerves, like you were going to go to a collateral ganglion, but there is no collateral ganglion there. What's there is the adrenal medulla. Okay, so we have a preganglionic neuron right here, kind of going like this, but it goes to the adrenal medulla. So the adrenal medulla. There's our adrenal medulla right there. And it communicates directly with the adrenal medulla. Okay? From there, we're going to be releasing chemicals into the blood, and the blood is going to carry those chemicals to the target tissue. Okay, so what we're going to have here is we're going to have a blood vessel right here, kind of leading off in this direction like this, and it's going to carry those chemicals to the target tissue. We'll talk about the significance of that here in just a couple of minutes, but for right now, we'll just go ahead and kind of outline the adrenal sympathetic pathway, because it is a little bit different than what we've been seeing with the other pathways. Okay, so we kind of have all of this outlined right now, where we have our sort of wiring diagram for each of our 4 pathways set up. We have somatic motor pathways, we have parasympathetic motor, sympathetic motor, and then adrenal sympathetic pathways sort of set up. Are there any questions about any of these wiring diagrams? And remember, we know that we are going to have a little bit more here because we've got to deal with the sympathetic chain on both of these, and that's why we're practicing that in lab. But this is a very basic arrangement of neurons. Okay, cool. Okay, so what we have here then is we have a situation where this neuron needs to communicate with this muscle, right? We have a situation here where this neuron needs to communicate with this neuron and needs to tell this neuron to do something, and then this neuron needs to tell the target tissue to do something, right? Same thing here. This neuron needs to tell this neuron to do something, and this neuron needs to tell the target tissue to do something. And then this neuron has to be able to tell the adrenal medulla to release this chemical that we'll talk about into the bloodstream so that it can be carried to the target tissue. Okay, so we have a lot of cell-to-cell communication that we have to start to think about here, and you guys have already talked about a little bit of it. So we're going to start with the somatic motor pathway. I'm going to ask you to dig deep into your memory banks, and let's talk a little bit about how a somatic motor neuron might communicate with a muscle cell to tell it to contract. What do we remember about that? How does this somatic motor neuron tell this muscle cell to contract? Releases acetylcholine, right? Releases acetylcholine, right? What type of chemical is acetylcholine? What do we classify that as? Do you remember? It's a neurotransmitter, yeah. So there are lots of different neurotransmitters in our body. Acetylcholine is just one of them. It does happen to be one that we talk about an awful lot. But acetylcholine is a neurotransmitter, right? So we have acetylcholine. Usually we just abbreviate it though as ACH. That tends to be a little bit easier than trying to write out acetylcholine every single time. Okay. So we learned in 50A that acetylcholine binds to receptors on the skeletal muscle. That starts an action potential in the skeletal muscle cell, and as a result of that, myosin and actin slide past one another and the muscle cell contracts, right? So acetylcholine binds to receptors called cholinergic receptors. Okay, that's the family of receptors that accepts acetylcholine. Okay, so it binds to what we call cholinergic receptors. Those names kind of sound the same. Acetylcholine, cholinergic kind of sound the same, right? Binds to cholinergic receptors. These receptors are just proteins, right? So they're coded for in our DNA, and some proteins can vary just a little bit here and there. So as far as cholinergic receptors go, there's actually quite a few that we've characterized and stuff like that. But for the sake of a class like this, we really only need to focus on 2 sort of main types of cholinergic receptors, okay? So there are 2 main types of cholinergic receptors that we're interested in. One of them is a type of receptor called a nicotinic receptor. It's called a nicotinic receptor. The other one is called a muscarinic receptor. So we have 2 types of cholinergic receptors. Sept, acetylcholine, we have nicotinic, and we have muscarinic receptors. If one of those names looks vaguely familiar to you, it should. nicotinic receptors are named because the molecule nicotine also happens to bind very, very tightly to these receptors. And in certain studies and things like that, if we wanted to study these receptors, researchers will often use nicotine to fill those binding sites really, really quickly. Nicotine has a shape similar to acetylcholine, and it just binds and very tightly to those receptors. So they get their name because nicotine does bind very tightly to those receptors. Just so happens there's another molecule or another chemical called muscarine that happens to bind very closely to these guys. You probably haven't heard of it, but it binds really tightly to those ones. So we use it when we study these receptors because they bind so tightly. Okay, so why do we have two receptor types and why do we care? Okay, well, the type of receptor that a particular tissue has is in a lot of ways going to determine the type of response that tissue has to acetylcholine. Okay, so we're going to make a really broad generalization, and if you really wanted to, you could probably poke a lot of holes in this generalization. But it's going to help us to remember this, okay? And I'd rather help us to remember it now and poke holes in it later than poke holes in it now and have trouble remembering things. So, the broad generalization that we're going to make is that when acetylcholine binds to nicotinic receptors, it tends to excite or activate a particular tissue, okay? So if a tissue has nicotinic receptors, acetylcholine binds to it, that tissue is going to be upregulated. It's going to be excited in response to acetylcholine, okay? If acetylcholine binds to a tissue that has muscarinic receptors, it's going to do essentially the opposite. It's going to down-regulate that tissue, okay? Again, this is a generalization. There's a lot kind of wrong with what I just said, but it's going to help us remember things. And you'll see why in just a minute, okay? Yeah?
Say that again.
Acetylcholine tends to excite tissues when it binds to nicotinic receptors and tends to depress tissues when it binds to muscarinic receptors. Okay. So, with that in mind, again, this is our wrong generalization, but we're going to run with it for right now. With that in mind, when we know that acetylcholine is released from this motor neuron, and it binds to receptors on the skeletal muscle, right? What does the skeletal muscle do in response to that? Contracts. Would you say that's exciting the skeletal muscle cell, upregulating the skeletal muscle cell, making the skeletal muscle do something? Yeah, I would too. So what type of receptor are we looking at here? We're looking at nicotinic receptors. Very good, right? Skeletal muscle does indeed have nicotinic receptors. Okay? Okay, we're good with that? All right, let's run through some of these then. Because we have a lot of cells that got to communicate with each other, and we're already at 8:26 a.m. So we got a lot of work to do. Okay, let's take a look at a sympathetic motor pathway. Well, we'll go in order. Let's take a look at a parasympathetic motor pathway. All right. So parasympathetic motor pathway, preganglionic neuron. Action potential, right? Helps if you make the sound. All the way to the axon terminal. Now, this neuron has to excite this neuron, okay? This preganglionic parasympathetic neuron is going to release acetylcholine, okay? So acetylcholine is released here as well. It's releasing acetylcholine onto the postganglionic parasympathetic neuron. Okay. Now, if we want to get the message to the target tissue, that means that the action potential that was in this cell has to get transferred to the action potential, or has to transfer an action potential to this cell, right? Because we got to eventually get the message over here to the target. So if we're releasing acetylcholine, we have to excite or stimulate this postganglionic neuron. What type of receptor do you think that this postganglionic neuron has? Nicotinic. Very good, right? Our postganglionic parasympathetic neurons have nicotinic acetylcholine receptors because that neuron is going to start an action potential in response to the stimulation from the preganglionic. Okay? You guys got this, all right? Let's go one more, okay? So now we started that action potential in our postganglionic parasympathetic neuron. And shorter, all the way to our axon terminal. We're going to release acetylcholine again, okay? This is an entirely acetylcholine system. So we release acetylcholine again onto our target tissue. What do we call our parasympathetic branch of our autonomic nervous system? I'm going to have a name for it. I think I might have used it last week. Your book certainly uses it. If our sympathetic nervous system is what we consider to be our fight or flight response, what do we call our parasympathetic nervous system? Rest and digest, right? So what I think sometimes when we think about our parasympathetic nervous system, we think about down regulating our tissues, right? Kind of, sort of. Is that what you think about? That's what I think about. Okay. So if I want to down regulate my tissues and I have acetylcholine as my neurotransmitter, what type of receptor do you think it's going to bind to? Muscarinic. Very good, right? It's going to bind to muscarinic receptors, okay? So our organs, our internal organs, have acetylcholine receptors, but they're muscarinic receptors, okay? They're muscarinic receptors. Okay? So we find nicotinic receptors on postganglionic neurons. We find nicotinic receptors on skeletal muscle, but when we get to our internal organs, the kind of targets that we see as part of the autonomic nervous system, those are muscarinic receptors, okay? So, we'll give a couple of examples here. Skeletal muscle. We have skeletal muscle, we have postganglionic neurons, and then this is internal organs. Okay, this is internal organs. Okay. Cool beans. All right, let's go ahead and take a look at our sympathetic motor pathway really quickly. Let's go ahead and start with our preganglionic sympathetic neuron. Action potential. Neurotransmitter released. It's acetylcholine, okay? Acetylcholine, okay? Acetylcholine is the name of the game here if you haven't figured it out yet. What type of receptor do you think acetylcholine is binding to on the postganglionic sympathetic neuron? Nicotinic, very good, okay? Because it's exciting that neuron, it's telling that neuron to do something, okay? Action potential here. And we get to the target. Now the story changes a little bit, okay? Can't be acetylcholine, okay? Because our internal organs have muscarinic receptors. Acetylcholine binds to those. It's going to do with the parasympathetic nervous system. We need another neurotransmitter. We need another set of instructions, okay? The neurotransmitter that we're interested in here, the neurotransmitter that we're looking at, is the neurotransmitter norepinephrine. I'm just going to put NE right here. I'm going to come over here. We'll put nor E or NE. It is our abbreviation for norepinephrine. Okay. There are two neurotransmitters that we're actually interested in this class. Norepinephrine is the first one. The other one that we're interested in is epinephrine. Very similar molecular structures. Probably know these kind of colloquially as noradrenaline and adrenaline, right? But their actual names are norepinephrine and epinephrine. Okay? Collectively, we call norepinephrine and epinephrine, we call them the catecholamines. Call them the catecholamines. So we can talk about catecholamine levels in the blood and stuff like that. Catecholamines are derived, norepinephrine and epinephrine are derived from a single amino acid. So that's where that ending of that word comes from, amine, because this guy's derived from a single amino acid. This is going to become important in just a couple of minutes when we talk about how these guys are broken down. But they're the catecholamines. But these post-ganglionic sympathetic neurons release norepinephrine onto the target tissue. Okay? Epinephrine and norepinephrine bind to receptors called adrenergic receptors. Bind to what we call adrenergic receptors. Again, big family of proteins, lots and lots and lots of them have been identified. But we're only interested in two primary ones. There are two primary ones. There's alpha and beta receptors. Okay? So if you've been in Southern California long enough, you probably remember the grocery store by the same name. You may never look at it again. Alpha, beta, right? Wasn't that a grocery store back in like the 80s in Southern California? You're like, that's too far. I don't remember that far back. I'm like, that was like last year, you guys. Come on. Alpha, beta, yeah, that was a grocery store, right? Okay, I didn't live in Southern California at that point, but I've heard rumors. But nevertheless, alpha and beta receptors are what we're looking at here. Now again, there's a whole bunch of these guys. There's like alpha 1, alpha 2, beta 1, beta 2, blah, 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 blah. We don't really care about all of that. I really only want to pay attention to the two, okay, alpha and beta receptors. Generally speaking, norepinephrine likes alpha receptors a little bit better. It can bind to both, but it likes alpha receptors a little bit better. So we'll say mostly norepinephrine here. And then in the same vein, epinephrine likes beta receptors a little bit more. It binds to them a little bit more tightly. It can bind to both, but it likes beta receptors just a little bit more. And again, this is going to be important in just about 10 or 15 minutes, so bear with me for a minute. But nevertheless, we have a couple of different types of receptors. Norepinephrine and epinephrine can bind to both, but they both kind of have their preference as well. Okay, so anyway, norepinephrine is released. It binds to receptors on the target tissue. All is good, okay? And it has its upregulatory effects. Are there any questions so far? So it leaves us with one final pathway. It's the one that we introduced to you guys this morning, the adrenal sympathetic pathway. This one is our very last one, and it's going to be very similar to what we've already talked about. So let's go ahead and run through this. Let's talk about what's happening here, and then what we'll do is we'll talk a little bit about some of the other aspects of this nervous system. So as far as the CNS goes, again, we're looking at T1 to L2, no different than this one. And we're looking at a preganglionic neuron that leads to the adrenal medulla, okay? So remember, there is no postganglionic neuron in the system, and that's kind of a curious thing, right? Like, why would there not be one here? when you look at the adrenal medulla under the microscope, it looks like a whole bunch of neuronal cell bodies. It's not. The cells that make up the adrenal medulla, we'll talk about them a little bit more next week, but the cells that make up the adrenal medulla are these big round cells with big visible nuclei in them. They look really similar to neuronal cell bodies. So we believe that probably somewhere in our distant evolutionary past, this was a ganglion of neurons that has evolved and lost those neuronal capabilities. But it still releases neurotransmitters in the same way that neurons would do. So it's just sort of an evolved neuronal ganglia that we have kind of hanging out on top of our kidney is kind of the belief. But nevertheless, these preganglionic neurons are going to lead to the adrenal medulla, and they're going to release epinephrine onto the adrenal medulla, or I'm sorry, acetylcholine onto the adrenal medulla. I'm getting ahead of myself here. So we're going to release acetylcholine onto the adrenal medulla. Okay. The adrenal medulla, in turn, is going to release these chemical messengers directly into the blood. The primary chemical messenger that the adrenal medulla releases is epinephrine, directly into the bloodstream. Epinephrine. There is a little tiny bit of norepinephrine that it releases, but it's not a whole lot in comparison. It's mostly epinephrine. Put a couple of molecules of norepinephrine to indicate that it's not as much, right? So we have epinephrine and we have norepinephrine being released into the bloodstream. Those neurotransmitters are then carried to their target tissues, and we have a response as a result of that. Okay. So why do we have two different sympathetic nervous system pathways, right? Why are there two different ones? Why do we have this one, which is very similar to the parasympathetic in a lot of ways, and then we have this one, which is entirely wackadoodle and different, right? Why are there two? I'm not going to really go through every single organ system and make you guys look at what the sympathetic and parasympathetic do to every organ system, right? We don't need to make a list of that. But it would probably not hurt to take a look at it, just kind of for your own knowledge. There's a really nice table in your book that shows everything that both of these nervous systems go to and the effect that they have. But let's pick an example for a minute. Let's think about your eyes and the pupils of your eyes, right? Your eyes are innervated by both sympathetic and parasympathetic fibers, and they're responsible for changing the diameter of your pupil when you go into changing light conditions, right? So when you go into a dark movie theater, for instance, your pupils are going to dilate, okay? That dilation of your pupils is the result of sympathetic innervation, okay? Norepinephrine was released onto those muscles, and it caused those muscles to contract and cause the dilation of your pupils, okay? When the movie's over, you go out into the bright sunlight, and in order to accommodate for that changing light, your pupils are going to constrict, right? We're going to see the constriction of your pupils as a result of that. That's parasympathetic innervation, all right? That parasympathetic is a result of acetylcholine being released onto the muscles that control those, that action in your pupils, and you see the constriction of your pupils, okay? I said this on Friday, I'm going to say it again right now. Sympathetic and parasympathetic innervation are commonplace in all of your internal organs, and they work to regulate homeostasis, okay? When we think of the sympathetic nervous system, we think of like that feeling that you get when you narrowly miss being in a car accident or something like that, right? When you get the bill in the mail that you weren't expecting to get, and you get that rush of adrenaline, like, I think we've all been there at some point, right? that's your adrenal sympathetic pathway at work, okay? Those short-term, intense stress situations, that's when the adrenal sympathetic pathway is stimulated, okay? And if you think about it, it makes a little bit of sense, right? When my pupils dilate when I go into a dark room, that's the only effect that I have, right? You don't feel that rush of adrenaline like you feel, or that rush of epinephrine like you feel when you narrowly miss being in a car accident. When you narrowly miss being in a car accident, you feel that rush of epinephrine everywhere, right? Your skeletal muscles, your pupils are dilated, your heart rate increases, your breathing rate increases. That's because that neurotransmitter is being released into the bloodstream. Okay, and the blood carries that epinephrine everywhere in the body. So that means that every place that has a blood supply, which is everywhere in your body, every place that has a blood supply is responding to that epinephrine. So it's like a global sympathetic response. Okay, everything is responding to it. When you have just normal day-to-day regulation of your body, you're able to stimulate here just very specific organs like the pupils of your eye, right? So there's a very different response that we see between the two, okay? Yeah. Exercise is not adrenal, it is sympathetic, but it's more global. A lot of sympathetic stimulation happening during exercise, but it's not adrenal. Okay. Any questions so far? Okay. So this diagram that we have on the board right now is drawn out in your lab. It's drawn out in your book. We use this a lot to sort of compare all of the different things, and it's a really good starting point for making comparisons. Even if it's a diagram that doesn't necessarily have all of this in it, you can still go in and you can plug in neurotransmitters. You can plug in the types of receptors that they're binding to. And I recommend taking that kind of whole approach to this so that you can paint a complete picture of sort of what's happening here. Okay. All right. If there's no other questions, let's go ahead and move on. We want to talk a little bit about some of the anatomy of these. Well, we've seen neuromuscular junctions here. I want to take a look at the junctions between these postganglionic neurons and the target tissue. So let's go ahead. I'm going to make myself a little bit of room here on the board. Let's go ahead and kind of take a look at that. So I'm going to shorten my blood vessel here just a little bit, and we'll kind of go from there. So what I'd like to do is I'd like to kind of take a look at this area right here. So if we take this and we sort of blow it up, I want to go ahead and kind of take a look at what we have right there. Again, we've sort of seen this one up close. You guys looked at the motor end plates and things like that in 50A regarding skeletal muscle. But I want to look at the relationship that one of these postganglionic neurons has with the target tissue. So let's go ahead and take a look at that really quickly. So if we blow that up, then you have the axon here of my postganglionic neuron. The axon of the postganglionic neuron. And then you've got your target. I think I'm just going to make some smooth muscle cells here, just for ease. But we've got our smooth muscle cells here. And remember, smooth muscles have this sort of fusiform shape to them. They're very long and slender at both ends. They kind of look like this, just kind of like this. Something along those lines. Another one over here maybe. So we've got some smooth muscle here. nuclei, make them look like proper cells instead of fish. There we go. So we've got our smooth muscle right here. All right. So what does the quote unquote neuromuscular junction look like here with this postganglionic neuron and then this target tissue, which in this case happens to be smooth muscle? So you guys are used to a fairly distinct axon terminal, and that's kind of what we've learned about so far. These postganglionic autonomic neurons are a little bit different. What they do is they tend to branch out fairly significantly, and what they do is they kind of create a web that goes over the entire tissue. So they have this web that kind of goes over the tissue like this. Your book has some good diagrams. I know I'm not the best artist on earth, but these are like the golf clubs sticking out of the yuccas. But we've got this sort of a situation. And then on those extensions, you have little bulbs that have the neurotransmitter in them, right? So we have these little bulbs that have the neurotransmitter in them, kind of like this. Kind of like that is what it looks like. These little bulbs are called varicosities. They have a funny name. They're called varicosities. Varicosities are what they're called. They look kind of like pearls on a necklace might look or something like that when you look at them underneath the microscope. So it's in those varicosities that we're actually synthesizing the neurotransmitter. So if it was a parasympathetic neuron, for instance, that's where the acetylcholine would be made. If it was a sympathetic neuron, that's where the norepinephrine would be made, right? So we're making the neurotransmitter here. The neurotransmitter is being synthesized in these varicosities. And that neurotransmitter is released. There's a couple of different things that can happen to it. One of the things that can happen is that it can bind to its receptor on the target tissue, which is what we've been talking about. But there's a couple of other things that we see that could happen. So when that neurotransmitter is released, it can bind to its receptor. So neurotransmitter can bind to its receptor. Which is the most obvious thing. That's what we've been talking about, right? And we know what happens when it does that. Another thing that can happen is that it can miss the receptor entirely and it can actually diffuse away. I think one of the things that we do is we often give like human qualities to these molecules, right? So I think sometimes we teach this and try to make you guys learn this from a point of view of, oh, the neurotransmitter is released and it goes to look for its receptor. It doesn't go look for anything, guys. It just like diffuses away. And the hope is that some of it lands in the receptor, but that's kind of all it is it's really just a big game of chance, right? I mean, that's all it is. So in order to make it work, we can help the chances by keeping the two things really, really, really close to each other, and also having a lot of receptors and a lot of neurotransmitter. That's the only way to really increase our odds. But really, it's just a game of chance, right? And I think we forget that sometimes, that like, all of our biochemistry is just luck that it even happens. But we're here, so we got lucky. So at any rate, it's an important thing to remember, right? So what we're doing is we're kind of flooding this whole area with neurotransmitter. Some of it's going to bind to receptors, but some of it is just going to kind of drift away, right? It's going to drift away. And we have to get rid of it somehow when that happens, okay? So some of it just kind of diffuses away. I think when I realized that and started to understand that really biochemistry is just a game of chance, that really kind of changed my view of the way that we work. That and the idea that we can run out of room in cells is kind of mind-boggling to me. We'll talk about that another day. But it's kind of a crazy idea that space inside of cells is actually quite limited, and we have to make compromises about what's inside of them. But anyway, it can diffuse away. And then when it does diffuse away, it usually is going to be absorbed by surrounding cells, and then it can be recycled and it can be broken down. Okay, so that's kind of a number 3 right here. Is it can be absorbed by surrounding cells. And it has to be broken down. Even if it binds to its receptor, it eventually has to get broken down, right? We can't keep that neurotransmitter in the receptor forever. It has to go away at some point, right? So even if number 1, which is I guess the best case scenario, happens, it has to be broken down. So there's a couple of different ways in which it can get broken down, but it has to get destroyed at some point. It can't hang out in our body indefinitely. Let's talk about how these neurotransmitters are broken down, all right? And you may not care a whole lot, but I guarantee there's some clinical significance here, okay? So acetylcholine, you guys might have talked about this in 50A. Acetylcholine is broken down by an enzyme called acetylcholine esterase. Broken down by an enzyme called acetylcholine esterase, or ACHE. Here's what that enzyme is abbreviated as, acetylcholine esterase. Acetylcholine esterase breaks down acetylcholine into its component parts of acetic acid and choline. Epinephrine and norepinephrine okay. Remember how I said that these guys are derived from a single amino acid? Okay, so we call them the catecholamines, but because they are made from a single amino acid, we also call them monoamines. And there's a whole crap load of chemicals that are monoamines. These are not the only 2, but they're called monoamines. So epinephrine and norepinephrine then are broken down by an enzyme called monoamine oxidase, or MAO. Okay? They're broken down by an enzyme called MAO, monoamine oxidase. Okay? There's a lot of clinical significance of these, right? Because like I said, epinephrine and norepinephrine are not the only 2 monoamines in our body. There's a whole bunch of other things that are also derived from single amino acids that happen to be broken down by this enzyme monoamine oxidase. Some of those chemicals that are monoamines are things like serotonin and dopamine, which are really important neurotransmitters in our brain. Okay? So how do we also know about serotonin and dopamine? Does anybody know like what their role, what they're implicated in?
Question?
Serotonin and dopamine. Do you know what they do? Those are another example of monoamine neurotransmitters that we find in our brain.
Serotonin, the one that's for like sleeping and relaxing.
Okay, sleeping and relaxing.
Dopamine is pretty much the same, right?
Kinda, yeah. I mean, like, a lot of times, I think the current evidence is that they're definitely involved with relaxation, but there also is pretty good evidence to suggest that serotonin and dopamine are involved with happiness and stuff like that, right? We know that, like, a hug, for instance, can cause dopamine release and things like that, right? So, a couple of other neurotransmitters that are important that are also broken down by MAO, monoamine oxidase, okay? Some people suffer from a lack of certain monoamines like serotonin and dopamine, and it's believed that that lack of those neurotransmitters might impact mental health and well-being, right? Happiness. In other words, we think that some cases of depression might have to do with lower than normal levels of chemicals like serotonin and dopamine. So if you've got lower levels of those chemicals that are supposed to make you happy, we probably don't want to be breaking those chemicals down a whole lot, right? So we probably don't want this enzyme to be working really, really hard in our bodies because that's breaking down those chemicals that might make us happy. So one of the common classes of drugs that is often given to treat people with depression is a class of drug called an MAO inhibitor, or an MAOI. Have you guys heard of MAO inhibitors or MAOIs? There you go, right? Serotonin and dopamine are monoamines. We don't want to break them down, so we inhibit this enzyme from breaking down those monoamines, and that hopefully helps to keep us happy, right? At least that's generally the idea, is monoamines. Some of them might make us happy, so if we're not happy, we want to keep those things in our body as much as we can. So we inhibit this enzyme. So a monoamine oxidase inhibitor is a class of drug that's sometimes prescribed to help to combat depression. I think our understanding of depression and mental health issues has probably evolved a little bit. As I understand it, though, we still don't completely understand it. So to some degree, we know that if we give you a certain drug and it makes you happy, then maybe you should keep taking that drug. But we don't really know how antidepressants work entirely. At least that's the way that I understand it. But at least there's a logical explanation for MAO inhibitors. So we have that. All right. And then finally, just to reiterate one final thing, this branch, this adrenal sympathetic branch, is where we're dumping these chemicals into the blood. And it's the first time where we've seen chemicals being dumped into the blood instead of being delivered straight to their source, okay? This is also the basis of the entire endocrine system, chemicals traveling in our blood, and that's what we're going to transition to next. Okay, we'll probably start to talk about the endocrine system a little bit today in lab. I want to give you guys a little bit of time after your quiz to review our sympathetic nervous system pathways, and then what we'll do is we will start to talk about the endocrine system and stuff like that. But it's not